Welcome to this first video on DNA and cell genetics. In this one, I'd like to deal with the topic of the structure of DNA so that you have a really good understanding of how DNA works in a cell. We've already taken a look at the nucleic acids, DNA and RNA in the biochemicals playlist. And if you didn't get a chance to look at that one, maybe scroll through to the end of this one. I'll put a link up here at the end. You can go back and have a look at that one. That one's a really good foundation for understanding what we're going to be dealing with in this video. So let's get started on this. You have to think back to almost 100 years ago when people would look at DNA under a microscope. They recognized chromosomes. They didn't really understand the structure of DNA. When they see a cell undergoing mitosis, they see chromosomes that look like this. They'd be lining up at the equatorial plate and the centromere would break and they'd migrate to the opposite sides. And they start focusing in on exactly what that chemical was made out of. They recognize it's made out of phosphates, sugars, and some nitrogen-containing bases. That's all there is. Oh, there's a little bit of protein as well. Proteins called histones, which they wrap around, but these are the major components there. One of the researchers that was looking at this thing was a fellow by the name of Chargaff. Chargaff looked at the chromosomes in different kinds of cells. I mean, humans have got 23 chromosomes in their sex cells, so a total of 46 chromosomes. 23 pairs. But in contrast, a fruit fly has only got 4 chromosomes. And what Chargaff was doing is he's taking a look at these different chromosomes in different organisms, and he'd consider these nitrogenous bases, and he always found out that the amount of one that he called, or that was called adenine, was always equal to thymine. So the concentration of adenine was always equal to the concentration of thymine, and the other two were always in the same concentration as well. So guanine is always equal to cytosine. And that's as far as he could take it. So he didn't understand why. How could that possibly be so true in no matter which chromosomes they looked at? The suggestion is that they bonded to each other, but they couldn't understand how that happened in a molecule that all they could see it looked like this. So along comes, after Chargaff, Watson and Crick. And a lady by the name of Rosalind Franklin. We often forget about Rosalind Franklin. Unfortunately, she passed away before the final structure of DNA was figured out, and Watson and Crick were left, and they actually got the Nobel Prize in the 1950s for figuring out the structure. But Rosalind Franklin dealt with X-ray crystallography, so she was able to take DNA molecules and look at them on the end. So instead of looking at it from the side like this, she could look up a DNA molecule. And what she found out was that they were hollow. They're actually hollow. You could look right down the middle of this thing. And here's all the sugars and phosphates, and the bases are around here, and there's nothing in the middle of it. And Watson and Crick were working on this, and they said, wait a minute, the bases are located in here. They could find the nitrogenous components in there like that. And they suggested that these things are strands and they're held together in a structure like that. So what you have is a DNA molecule that it's a double-stranded molecule where the two strands wrap around each other in a helical fashion like this, wrapping it around, and the hydrogen bonds which hold bases together are like that. So this is all hydrogen bonded in here. These are just a pile of hydrogen bonds right down the middle of this structure. And they got the Nobel Prize for figuring out that structure. 1956 they got the Nobel Prize for it, although they figured out the structure in 1953. This is our topic in this video. What the heck is this thing and how does it really work? One of the things that I'm going to add to this right now is I'm going to do this. I'm going to put 3 little lines here and just an equal sign there. And I'll show you why I'm going to do that. I'll show you that. I'm going to hang up this picture which I've pre-drawn, and I can show you why in a second here. I'll just erase these people's names right here. These are the 4 bases right here. This is adenine, this is guanine, this is thymine, and this is cytosine. The names are there, but the abbreviations A, G, T, and C. Hydrogen bonded to each other. These numbers of lines there represent the numbers of hydrogen bonds. So what you have, if you look at it carefully, is a positive dipole there and a negative dipole here, a negative dipole and a positive dipole. That sets up one hydrogen bond right here and another one here. This negative dipole, positive dipole, you can see there's another hydrogen bond. A positive dipole, a negative dipole, another hydrogen bond, a positive dipole, and a negative dipole, another hydrogen bond. These lines here represent the numbers of hydrogen bonds between them. So what you have, if you take this structure here and you lay it out flat, so we've got a sugar, phosphate, sugar, phosphate. You've got a base, let's say this was adenine, nothing bonded to the phosphate. There's a base, let's say this one is guanine, nothing bonded to the phosphate. Of course this goes on and on, both directions. Adenine always bonds to thymine by 2 hydrogen bonds. So there's another sugar and another phosphate. I'm going to put this phosphate upside down. It looks like a D, but it's really a capital P. It's phosphate. The reason I'm going to do that is this strand is called anti-parallel, where that one goes that direction, this one goes the opposite direction. So it's really, S upside down looks like an S, so it doesn't matter for that one. And we can tell what letter's going to have to be there, can't we? Because guanine always bonds to cytosine, and there's always 3 hydrogen bonds there. So that's what DNA structure is like. But this is really, really, really long. I mean, we're talking about a huge, huge, huge macromolecule there. If we do this, I can just erase this and simplify things just a little bit. If here's one strand, I'm not going to put all the sugar phosphates on. I'll just go like this. And I'll put some bases on. Just a random selection of bases. You could tell me, I'm sure, what the bases on the other strand are. Because this is it has to be T, this one has to be a G, this one has to be C, an A, a G, a T, an A, a G, a C, an A, and a T. And they're hydrogen bonded together inside. These things here, because you always know the other one, if you know the first one, they're called complementary. They're complementary strands, and these are complementary bases. And we use the term complementary base pairing. Now that sets up another whole set of questions, I suppose, because, let me steal this space. If we go back and think about Chargaff's work for a second, and if Chargaff was studying Drosophila melanogaster's chromosomes, his fruit fly's chromosomes, he found that 23% were adenine. What's the percentage of guanine? If 23% of the bases were adenine, what's the percentage of guanine? Well, if we know that 23 is adenine, we also know that 23 must be thymine. The total of those two is 46. 100% minus 46 gives us 54. Half of them have to be guanine and half of them have to be cytosine. So 27% guanine and 27% cytosine. Now, is it possible that it could be wrong? Could it be a little bit out? Sure. I mean, if you think about it, A and G, adenine and guanine, they're the same size molecules. So theoretically, I suppose, you could take this one out. I won't take it out, I'll just cross it out. And adenine would fit there. It would fit perfectly fine. I mean, it's the same size thing. It wouldn't bond properly because adenine is supposed to bond to thymine. There's supposed to be 2 hydrogen bonds here. Adenine has these certain dipoles that are set up. This one over here, cytosine, has certain dipoles set up. So if you took this molecule and you shifted it up here, you can see the dipoles don't match. So it'll fit, but it doesn't bond together properly. This is what a DNA mutation is. That's an actual mutation. It's called a point mutation or substitution. That's a topic in the 4th video in this series. We're getting into all the ramifications of what happens in terms of the synthesis of proteins from there. So that sort of sets us up now. Let me take this down and go back to the original diagrams for DNA that we talk about all the time, these things that look like this. If this is a strand of DNA, we know that DNA has genes in it. So then what's a gene? Quite simply, a gene is a segment of DNA. This could be another gene over here. This could be another gene here. A DNA strand, one chromosome, has many genes, hundreds, sometimes thousands of genes on it. And that's how chromosomes determine protein synthesis. The gene actually determines the formation of a protein. And just flip back to this for a second. If this change that I put in here was part of a gene, then you'd have a mutation in the gene, a genetic mutation then, and you'd wind up with an incorrect protein being formed. That's where we're going with this topic. Before we do that though, before we end this video, we have to mention things like genotypes and phenotypes. We talk about phenotypes are physical appearances. Whether you have blue eyes or brown eyes or big ears or whatever, those are physical appearances. Those are the phenotypes. Phenotype is physical appearance. The genotype is the genetic makeup. And what you realize now from what I've just said is that the genotype, the genetic makeup, determines the phenotypes. So you are what you are because of your genes. You are tall, you are short, you have brown eyes or blue eyes because the genes make the proteins that allow you to have those physical appearances. And something else we have to mention too, when cells divide, as they do in mitosis during growth and repair of tissues, we wind up with, see, here's a parent cell here, and there's 46 chromosomes in it. We wind up with two cells, both with 46 chromosomes. Well, where'd the other 46 come from? The fact is, DNA has to be able to make copies of itself. That's the topic of DNA replication. So, and that's the topic of the second video. This goes on in cell division. The business of DNA replication, and then we'll deal with the business of protein formation. And then we'll zero in on DNA mutations. So, I think that summarizes the structure of DNA, gives you a little bit of an idea of the history of how it was formed, and we need to know all of this information moving forward. Thank you very much.